Hello everyone, I'm Priyanka and today we will discuss growth, nutrition and metabolism of bacteria. The minimum nutrition requirement for growth and multiplication of bacteria include source of carbon, nitrogen, hydrogen, oxygen and some inorganic salts. Further, we can classify bacteria on the basis of nutrition as follows. Autotrophs, these are the bacteria which can synthesize their essential metabolites, that is, their organic compounds, from the atmospheric carbon dioxide and nitrogen, are known as autotrophs or lithotrophs. Further, the autotrophs obtain their energy either photosynthetically, that are photolithotrophs or photoautotrophs, or by oxidation of chemical compounds. These are called chemolithotrophs or chemoautotrophs. Next, we have heterotrophs. These bacteria are unable to synthesize their own metabolites. They depend on performed organic compounds. Majority of pathogenic bacteria are heterotrophs and parasitic. They have evolved with the animal body where ready made nutrients are freely available. Next, we will talk about bacterial growth. First, we have bacterial cell division. So, the bacteria divides by binary fission. When a bacterial cell reaches a critical mass in its cellular constituent, the cell division starts. Bacterial nucleus or chromosome is a circular double stranded DNA molecule. The nucleus division precedes bacterial cell division. During replication, the two strands of DNA are separated and new complementary strands are formed. Thus, the two identical double stranded DNA molecules are formed. As you can see, the process of binary fission in figure 3.1, the replicated DNA molecule are distributed to the two daughter cells. The transverse septum grow across the cell from the cell membrane. This is followed by the deposition of the cell wall material and two daughter cells separate. In some bacterial species, the daughter cell may remain partially attached even after cell division. Talking about the clinical significance, some bacteria are present as uh, streptococcus or streptococcus, that is in a chain, or diplococcus. Morphology of diplococcus and in chain are due to non-separation of daughter cell after cell division. मतलब सिंपली जो डॉटर सेल सेपरेट आउट हो जाते थे, मतलब जो सेपरेट आउट हो जाते थे, लेकिन कुछ केसेस में क्या होता है वो पार्शली अटैच रहते हैं आफ्टर सेल डिवीज़न भी. सो ये उनकी जो कैरेक्टरिस्टिक प्रॉपर्टी रहेगी, जो उनकी मॉर्फोलॉजी में रहेगा, वो उनको आइडेंटिफाई करने के लिए हेल्प करेगा. These morphological features are very useful in identification of these bacteria in laboratory. Next, we have generation time. The time required for the bacterium to give rise to two daughter cell under optimum condition is known as generation time. The generation time in most of the medically important bacteria is about 20 minutes. In Mycobacterium tuberculosis, it is about 20 hours, and in Lepra bacilli, it is 20 days. As bacteria grow so rapidly and by geometric progression, a single bacterium can theoretically give rise to 10 raised to power 21 progeny in 24 hours. In actual practice, bacterial multiplication is arrested after a few cell division due to exhaustion of nutrients and accumulation of toxic products. Talking about the clinical significance, when generation time is about 20 minutes, most of the medically important bacteria takes almost 18 to 24 hours to grow on culture media. In contrast, Mycobacterium tuberculosis takes longer duration in weeks to grow as its generation time is longer, that is 20 hours. Mycobacterium leprae takes months to grow in animal models as its generation time is 20 days. Next we have bacterial count. Total count. Now this total count indicates total number of bacteria in specimen irrespective of whether they are living or dead. Total count may be made by counting the bacteria under microscope using counting chamber. Next we have viable count. It measures the number of living, that is viable cells. Viable counts are obtained by dilution or plating method. In dilution method, several tubes with liquid culture media are inoculated with varying dilution of samples and the viable count calculated statistically from the number of the tubes showing growth. Through this method does not give Though this method does not give accurate value, but it is still used widely for finding out the presumptive coliform count in drinking water. In the plating method, on other hand, approximate dilutions of the specimen are inoculated on solid media, either on the surface of the plate or as pore plate. The number of the colony that grow after incubation give a estimate of the viable count. Each colony represents a clone of bacteria derived from a single parent cell. Therefore, one colony is equivalent to one bacterium. Number of colony are equivalent to number of bacteria in the specimen. Talking about this clinical significance, viable count is useful in clinical microbiology for testing of water by presumptive coliform count and also in the diagnosis of urinary tract infection. Next we have bacterial growth curve. When a bacterium is inoculated into a suitable culture, liquid medium and incubated, its growth follow a definite course. When bacterial cell of such culture is determined at different interval and plotted in relation to time, a growth curve is obtained. The growth curve has 4 phases. First we have lag phase. After inoculation of culture medium, multiplication usually does not begin immediately. The period between the inoculation and beginning of multiplication is known as lag phase. During this period, the organism adapts to the new environment, during which necessary enzyme and intermediate metabolites are built up in an adequate quantity for multiplication to proceed. There is increase in size of the cell, but there is no appreciable increase in number. The duration of the lag phase vary with the species, nature of the culture medium, temperature, etc. So this is all about lag phase. Next we have log phase, which is also known as exponential phase. The cell division start and their number increase exponentially or by geometric progression with time. If logarithm of a viable cell is plotted against time, a straight line is obtained. Next we have stationary phase. After the log phase, the bacteria growth eases almost completely due to exhaustion of nutrients and accumulation of toxic products. The number of the progeny cells formed is just enough to replace the number of cells that die. The number of the viable cells remains stationary as there is almost a balance between dying cell and newly formed cell. Next we have phase of decline or death phase. After a period of stationary phase, the bacterial population decreases due to death of cell. The decline phase started due to exhaustion of nutrients, accumulation of toxic products and autolytic enzymes. There is a decline in viable count and not in total count. With autolytic bacteria, even the total count show a phase of decline. Next we have morphological and physiological alteration of cell during growth curve. The end phase of the lag phase, bacteria have maximum cell size. In the log phase, cells are small and stain ununiformly. In the stationary phase, cells are gram variable and show irregular staining due to intercellular storage granules. Sporulation occurs. Many bacteria produce exotoxin. In the phase of decline, evolution forms are common. So that's all we have in the first part. Thank you and have a nice day.